thank you all for coming to my talk today. So I'm going to present some early data from my dissertation, and while this is early data, I think it's a great starting point to build from. I'm going to be talking about propagules, and so in red mangroves, they produce these structures that are actually viviparous. So they have fruits which germinate while still attached to the parent plant, and so the unit of dispersal is actually this structure that's more or less a young seedling rather than a fruit or seed. And so I developed this project under the context of a bit of theory that I think, even though it's somewhat controversial, I think is still worth exploring. So with the advent of next-gen sequencing technology, we've come to realize that microbes are more ubiquitous, diverse, and functionally important than previously thought. And so with this increased knowledge, we've begun to appreciate the diverse roles that microbes are playing in the physiology of multicellular organisms. And so my work is mostly interested in this assumption right here, and in investigating the degree to which endophytic communities are derived either from the parent of the multicellular organism or acquired from the environment. And so that's what I'm going to talk about today. I'm not going to answer that today, but that's sort of the guiding context for my dissertation work. So I'm exploring this in this system using the species Rhizophora mangle, which is commonly known as red mangroves. And so red mangroves are found throughout the tropics and subtropics in the Americas, as well as the western coast of Africa. And they provide numerous ecosystem services, like all forests and grasslands and vegetated areas, but they're unique in providing a sort of natural buffer between landward ecosystems and seaward ecosystems. So they protect inland ecosystems from things like storm surge and sea level rise, while protecting things like coral reefs and seagrass beds from getting silted over. Despite that, they face massive amounts of anthropogenic destruction, which undermines the resilience of coastal areas to things like sea level rise and climate change. Red mangroves are particularly well adapted to long-distance water-based dispersal, and so this is playing a role in their ongoing range expansion into the Florida Panhandle. Also, this dispersal vector, I think, makes this a good system for looking at potential inheritance of microbial communities, because in this situation where the propagule needs to survive potentially a long time in seawater and then survive a potentially stressful establishment environment, that may act as a selective filter where coming sort of preloaded with the microbial endophytes may be more beneficial in such a situation. And so the main question I'm going to talk about today is just how does this bacterial diversity vary across geographic distance? And as I mentioned, this is sort of a building block to answering some of the more complex questions that I'm trying to answer with the rest of my dissertation. So this is pretty straightforward. Just I wanted to show you guys where most of my field sampling is occurring. So most of my sites you can see are clustered in the Big Bend region of Florida. Just want to point out, I have these red pins which are representing, wild mangroves growing in their natural habitat, and then the green pins are representing two populations that I was fortunate enough to sample from. They're growing in nursery or greenhouse conditions. So up here, you can barely see this one because it's kind of hiding, but this is from the Gulf Specimen Marine Lab, which is sort of a outreach aquarium type institution. They were fortunate or kind enough to let me sample their mangroves. And then down here, this is a really unique population that I sampled from a company called Mangear, which essentially they sell outdoor apparel and then plant mangroves with the proceeds. But their setup for growing mangroves is actually it's not on the coast, it's inland in sort of these artificial flats. So it's a really unique population compared to some of these wild populations. You can also see it's the only one from South Florida and the only one that's on the Atlantic coast. So here you can see the exact moment that I realized I was underprepared for this field work. This is from my first trip into the field trying to work out my sampling methods. Since then, you can see the vest and the long sleeves. I've come a long way. and worked out a really nice and effective means of collecting my samples. So essentially, I am collecting tissue and placing my samples in sterile bags, and each sample is consisting of 10 propagules or 10 leaves collected at random across an individual. And so I wear gloves and sterilize my hands between each sample. The samples are then put on ice and kept on ice until I can bring them back to the lab and freeze them. So I'm going to talk about two different approaches I took in the lab and sort of the complementary results from them. So my main focus from the beginning has been using this next generation sequencing, using the 16S rRNA gene, which is sort of a really common marker for these types of studies. And despite the name, so this is actually DNA that I'm sequencing, but it codes for the ribosomal RNA structure. So just not to be confused there. And then as I was trying to get this off the ground, I was running into some issues. So I also did a second project where I saw what could be cultured from the propagules, because again, sometimes getting the next gen sequencing, making sure that you have enough DNA, isn't always as straightforward as you would like. But both of these approaches start in the same way. So I begin by taking my samples and adding phosphate buffer solution, and then essentially pulverizing them in this machine, which is known as a stomacher machine. Basically just consists of two paddles that just obliterate your tissue to something like that. I then take the liquid and then go in two different directions with it. So for next-gen sequencing, I use the liquid to then begin DNA extraction, and then for the culture-based methods, I use that liquid then to start culturing the microbes on solid media and kind of go from there. So for the next-gen sequencing, this is a very basic pipeline, and if you want to know more, I'd be happy to go into more detail afterward. But essentially, again, I'm starting collecting samples, extracting DNA, and then feeding them into this Illumina-MiSeq sequencing pipeline. And then all of my bioinformatics was done in the program QIIME2, which is again sort of the classic program for analyzing these types of data sets. The types of samples I have, as you can see, are almost all Rhizophora mangle leaf samples. And so again, as I was in the process of getting this pipeline working, I was having some difficulty getting I'm calling them fruits here, I apologize, I should say propagules. But I was having some difficulty getting the DNA getting sufficient DNA to be able to describe the communities in those tissues. So I decided to focus on leaf samples for this talk. And I also included samples from a couple of different species of mangroves. So these are white mangroves and black mangroves. Because in some of my future work, I'm going to be looking more closely at differences in microbial communities across multiple origins of mangrove habit. And so I wanted to make sure that I could work out the pipeline, not just for Rhizophora mangle and for the leaves and the propagules, but also for these different species and for various tissue types. So jumping back to culture-based sequencing, again, I essentially saw what could be cultured from that liquid, and then used just Sanger sequencing of the 16S region again, and then use the NCBI blast tool to identify what microbes I was finding. And so I found 4 different bacterial taxa using these methods, and I think these 2 are particularly interesting because these have been well described as plant endophytes before. Especially Pantoia mantis is particularly interesting because it's something that's been described across the board as something, everything from a plant pathogen to a harmless commensalist, and even a beneficial endophyte in some cases. And so I think that's interesting because a lot of times we like to have kind of neat bins that we can put things in. But in the case of these microbes, sometimes the environmental context is probably going to be determining what the microbe is actually doing. And so these 2 taxa, you might recognize these as human pathogens. And so there's, my first thought was, well, those are probably just contaminants. But they are actually also in the literature as having been isolated from plant tissues previously as well. So in comparison, though, the slide's a little busy, so bear with me for just a moment. But these tables are from 3 different samples of Rhizophora mangle propagules. And these are ones that I got to work with the next-gen sequencing. So these are showing the top 7 or 8 genera of microbes found within those tissues. And what's really interesting here is that none of the 4 taxa that I got from culture-based sequencing are in the top 7 or top 8. I say top 7, the top 8 includes the unclassified genus level. So really it's the top 7. So none of the culture-based taxa are showing up in the top 7. And we actually get some pretty decent similarity. So each of these colors is corresponding to a specific taxa. If you can't read that in the back, don't worry. I'm not going to go into detail about these specific taxa. But I just think it's interesting that we're recovering a lot of the same diversity across different samples. And what makes this even cooler is that these are coming from the same location. That's why T stands for Yankeetown. It's the same location that I got the propagules that I used for the culture-based sequencing. So this isn't necessarily surprising, but it's cool that next-gen sequencing is going to reveal different diversity than culture-based sequencing. And that's something that's sort of well established, that a lot of times what we're able to culture are really just the weeds of the microbial world. They're never going to grow easily. And so we need to use these next generation methods to really capture a better idea of what diversity is there. So to dive in a little deeper with this, this is a principal correspondence analysis, and what this is showing is each of these dots is one sample, and they're color-coded based on locality or geographic origin. And so you can see a nice cluster here of the Mangrove samples, which again are these ones down here. And again, that's kind of not surprising that they're distinct from everywhere else, because again, they're the furthest away geographically, they're grown in the most artificial environment, and they're also on sort of the southeast coast. But what's also kind of cool here, and I'll show you on the next page that there are actually significant differences between these, but if you look at the Cedar Key samples here, Cedar Key is actually between Yankeetown and St. George, so Cedar Key is further north than Yankeetown, but yet it's more different from these northern populations than Yankeetown is. And so I think that's interesting because it suggests that these microbial community composition is not just dependent on latitude or geographic origin. So briefly here, this is a pairwise permanova that shows significant differences between localities. So each locality is significantly different from each other potential combination. And so you can just kind of focus on the p-values here, but this is based on the taxonomic composition of these communities. And so we are recovering significant differences between them. So in conclusion, next-gen sequencing and culture-based sequencing are revealing different diversity. They're able to show different things. And it appears that endophytic communities are significantly different based on locality, but the question still remains, why is that? And so in the rest of my dissertation, I hope to sort of tease apart the influence of environment and phylogeny on these endophytic communities. I'm also going to be doing some work looking at differences in endophytic diversity across these multiple origins of mangrove habit. And this is sort of a sub-question of that. And then last but not least, if you read my abstract, you might have been expecting a little more phylogeography and rad-seq, and unfortunately that didn't quite pan out, but that is something that I'm still looking into, and how this species is expanding into the panhandle. So with that, I'd like to acknowledge my funding sources, my advisor, and all the people who have helped me along this path, and I would be happy to take any questions.